Good day, ladies and gentlemen. Today we are going to look at functions of the cell membrane and the organelles. So in this lecture video, you are going to learn number 1, types and functions of organelles. 2, you are going to learn the functions of plasma membranes. 3, you are going to learn endocytosis and exocytosis. So at the end of this lesson, you are expected to be able to describe the different organelles that are found in the cell, and you should be able to give examples and explain the functions of each of the organelles. You are also required at the end of this lesson to be able to describe plasma membrane, at least the function of the plasma membrane. And lastly, you are expected to be able to list the factors that regulate the movement of materials across the plasma membrane. So let's look at the functions individually. Okay, now let's look at organelles and their functions. Number 1, we have the rough endoplasmic reticulum, which functions in the synthesis of protein due to the presence of ribosomes, and also it undergoes another function of degradation of worn out organelles. So rough endoplasmic reticulum, you know we have endoplasmic reticulum is divided into 2. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So now the rough endoplasmic reticulum carries out the function of protein synthesis because it has ribosomes. And also it also carry out the function of degradation of worn out organelles. Smooth endoplasmic reticulum is involved in the synthesis of lipids and steroids, and also storage and metabolism of calcium and detoxification of toxic substances. So if you remember the structure of the cell, where you see the various organelles were labeled, if you look at this look like what we label as what the endoplasmic reticulum. So this is like a more clear representation or pictorial representation of the endoplasmic reticulum. It's like a kind of zoom picture of the endoplasmic reticulum, where it has various components attached to it. You have the smooth endoplasmic reticulum here. You can see it. It has nuclear envelope, and it has there is nuclear pore here, and it has what we call the cisternae, and it has, look at the cisternae here. It's like a kind of zigzag foldings that forms the cisternae, and in between there is space, which is the cisternal space. So this forms part of the rough endoplasmic reticulum, to which the ribosomes, you can see the ribosomes are attached or embedded in it. So this starts out function of what? The protein synthesis. Number 2, we have the Golgi apparatus, which functions in the processing, packaging, labeling, and delivery of proteins and lipids to their destination within the cell. So after the endoplasmic reticulum carries out its function of either be it the rough or the smooth, either maybe protein or the lipids is being produced. So it needs to be what? Processed and packaged appropriately and deliver into the appropriate destination within the cell. So Golgi apparatus carries out the function of that processing, packaging, and also delivery of the synthesized protein or lipids. So if you look at this structure, it is a more typical or clear representation of the Golgi apparatus. You can see from this part where you have this is the part that is having the endoplasmic reticulum, right? So if you look at this black cycle, it cycles part of the endoplasmic reticulum and the Golgi apparatus. So if you look at it now, this is like zoomed picture of what was cycled here. Look at it. This is part of the endoplasmic reticulum, and this is the Golgi apparatus. So they are closer. You know, there is what we call the cis or forming phase of the Golgi apparatus, which is closer to the rough endoplasmic reticulum. Look at it, and you have the trans or maturing phase, which is the other side. The other side, opposite, should I say, the other side that is facing the cell membrane. You know, on the inside, you have the cis or forming phase, which is close to the rough endoplasmic reticulum, while on the other side, you have the trans or maturing phase, which is close to the what? Look at it, close to the cell membrane. And you have also have these foldings of cystine A, and it have lumen. It has transport vesicles, you know, and you have the vesicles that are newly formed here. It has tubules, it has secretory vesicles and coated vesicles. This is the Golgian, forming Golgian vacuole that is being formed. So this is the typical structure of the Golgi apparatus. So don't forget the function is processing, packaging, and labeling, and also delivery of proteins and lipids to their destination within the cell. So let's look at the next organelle, which is the lysosome. The lysosome carries out the function of degradation of macromolecules, worn-out organelles, and also the removal of excess excretory products. It has the thickest membrane to prevent the leakage. So among the organelles, it's one of the organelles that have the thickest membrane that helps in prevention of leakage of hydrolytic enzymes. So this lysosome contains many hydrolytic enzymes, more than 40 different hydrolytic enzymes, and they are collectively known as lysozymes. So it has thick membrane that prevent these enzymes. Look at this. These are typical representation of the enzymes. This is the 2D representation of the lysozyme, and also this is the 3D representation of the lysosome. So it has the enzymes and proteins, and this is the thick membrane. So it's function in the degradation of worn-out organelles and removal of excess secretory products. So the next, we have the peroxisomes, which carries out the function of detoxification of hydrogen peroxide and other radicals because it contains antioxidant enzymes such as peroxidases and catalase. It carries out the function of breaking down excess fatty acids, degradation of purines to uric acid, and formation of bile acid. So this is the structure of peroxisome. If you look at it, this is now like the whole structure, and this is like transverse versus like cut into two. This is transverse section. You have the hydrolytic enzyme mixture. You have the matrix. It has also the membrane. Also, it has plasma membrane. Remember, we are talking about eukaryotic cell, and eukaryotic cell organelles have many of them have what membrane-bound organelles. So these organelles that we are now talking about. You see, they have a lot of them have what membrane. So this is plasma membrane and membrane transport protein, which is glycosylated. Here, this is in green that you can see, and you have also lipid layer because it has membrane. You should expect that membrane should consist of lipids and also protein. You know, you remember in our previous class we talked about plasma membrane, and in this also class we will also revise and give more function of the plasma membrane. So we have the mitochondria. Mitochondria, which is called the energy house of the cell. It's carried out the function of production of ATP, energy in form of ATP. You know, ATP is adenosine triphosphate. ATP is the energy currency of the cell, and this ATP is being produced from the citric acid cycle, which from basic biology, you know, Krebs cycle. But here in biochemistry, we mostly call it citric acid cycle or tricarboxylic acid cycle, which is called TCA cycle. So it is maternally inherited. It means it can be inherited from mother, and but absent in red blood cells. It's not present in red blood cells, and it's also function in the initiation of apoptosis. Apoptosis, remember, programmed cell death. So this is the structure, typical structure of mitochondria. It has also cystine, you know, that folding, zigzag foldings, and it has proline. Look at it. It also has some ribosome in it. It has granule. It also has DNA. So it's not only nucleus that has DNA. Mitochondria also have DNA. So you should take note that these two organelles, nucleus has DNA, and also mitochondria has genetic material, which is DNA. So you have ATP synthesis is taking place in the what? Inner mitochondrial membrane. So this is the inner membrane where ATP synthesis here is taking place. And you have the outer membrane. Look at the outer membrane, okay? And this is the what? Intermembrane space. And this is the matrix. So look at it. It's been labeled very well. So take note of the labelings. Okay, let's proceed. The next one is the ribosomes, which carry out the function of the protein synthesis occurring in the ribosomes. So we also have the nucleus, which it controls all the cellular activities, and absent in mature red blood cells. You remember, mitochondria is also absent in red blood cell. You see, it's absent in red blood cell, and also you have absent in mature red blood cell. You do not have what we call nucleus. Okay, so DNA and RNA synthesis and storage of genetic information is being carried out here in the nucleus. So this is the structure of nucleus. It has the nuclear membrane. Okay, it has the nuclear membrane, and it has the nuclear pore. This is the nuclear pore. It has ribosomes. Look at ribosomes. This is endoplasmic reticulum. Okay, and these are all ribosomes. Okay, it has the chromatin. It also has the nucleolus. Okay, and it also has the what? This is the nuclear envelope. So now let's look at plasma membrane and its function. We already know what plasma membrane is. Now let's look at the various function it carries out. So it carries out the function of protection. Plasma membrane protects the cytoplasm and organelles present in cytoplasm. It is responsible for the maintenance of the shape and size of cells. The most important function of the cell membrane is transportation, regulation of materials across the membrane. So this plasma membrane, the protection of it is that it serves as like a whole. If you have a city that is being fenced by a bigger wall, so that wall can serve as, analogically, can serve as the plasma membrane. It protects what comes in and what goes out of the cell. Okay. Now it carries out the function of transportation, transport of materials across the cell membrane. The cell membrane acts as semi-permeable membrane. It allows only some substance to pass through it and act as a barrier for other substances. So movement of substance has to be regulated. It's not everything that will come into the cell, and it's not everything that will just go out of the cell. It has to be what? Regulated. So that is what we call passive transport. Passive transport in cells involve the process of diffusion, and diffusion can be simple diffusion or facilitated diffusion. So in simple diffusion, we can say because of the cellular activities that is taking place, the rate of simple diffusion can be affected by temperature, number one. Number two, the molecular size, and also the concentration of the gradient. So materials that are moved through membranes by simple diffusion includes, take notes, have water, carbon dioxide, oxygen, and some lipid-soluble molecules such as alcohol. We can also have facilitated diffusion, where most molecules that cannot move freely through the membrane have to cross the membrane with the help of membrane transport proteins, which these transport proteins temporarily bind to the substances, and it's aids in the movement through the cell membrane, and this process is called the facilitated diffusion, or is also called passive transport. So let's look at this diagram. We can see active now and passive transport. For the active transport, it requires movement of molecules against the concentration gradient. So if we are moving molecules from a region of lower concentration to a region of higher concentration, you should expect that you need more work, more energy is going to be needed to carry out that function. For instance, if you want, let's give this example now. If let's say you have a class or a car that is already filled up, it is already full, the car is full, there is no space for anybody to enter. Now you want to introduce another passenger into the car. So you need to force that passenger, squeeze that passenger inside, inside, because it is against the concentration gradient. So there is, from a region of low concentration, only one passenger, and to a region of many passengers, a region of higher concentration. So you need to force that passenger to enter that car. So that is like that. So you need energy to do that, okay? So passive transport, you can have a transportation of oxygen. Oxygen can just diffuse through the membrane. You have extracellular fluid outside the cell, like oxygen can just enter inside directly. And you have like sodium also, ion can enter because it has sodium channel. This is aqueous channel. Now sodium can just enter by passive transport. It's proceed as diffusion from areas of what? High concentration, where I have them in high abundance here, into the area of low concentration here. So you should expect it to enter freely without use of any energy. So for the active transport, because it is against the concentration gradient, active transport proceeds against the concentration gradient and requires an input of energy. So example, sodium potassium ATPase, sodium potassium pump, okay, whereby you transport sodium, okay. When you transport sodium outside the cell, and also transporting potassium inside the cell, that requires ATP. So ATP has to be what? Hydrolyzed for that activity to effectively take place. So this requires energy. In essence, it requires energy, while passive does not require energy. So for facilitated diffusion, the process may be coupled to the movement of another molecule in the same direction or opposite direction. So we can say if facilitated diffusion requires movement of another molecule in the same or opposite direction, we call it co-transportation. Co-transportation. So in co-transport, the transport of one molecule depends on the sequential transfer of another molecule. So if you look at this picture, the first one that has A, B, A, B, A. So we have unipolar. This is a protein in the membrane that transports just a molecule in one direction. This is uniport. You have a symport carried out by a symporter. It means it carry out two molecules, transport two molecules at the same time in the same direction. So this is a subdivision of what transportation, which is carried out by symporter. And you can have antiport, a transportation of two molecules but in opposite direction. You can say this is counter transport. Antiport transport. It is being carried out by antiporter protein, okay? And if you look at the example here, you can see sodium glucose transporter is an example of protein that carry out that function of what SIMPOT, okay? While you can have antiporter sodium-potassium transporter can be an example of antiporter, where you have transport of sodium and potassium in different direction. While sodium is coming in, potassium is going out, okay? While here you can have both glucose and sodium going inside the cell. So we can see a SIMPOT moves two molecules in the same direction, example, sodium glucose transporter. While for antiport, we can say a system that moves two molecules in opposite direction, which is also known as counter transport, e.g. example is sodium potassium transporter, as we earlier explained. So let's continue. So now we have what we call endocytosis and exocytosis. So, you know, all cells need to move some substances through membrane in the same direction, or to counter in the same direction, counter to the gradient, or move substances that are too large or bulky with use of cell energy. Okay, we have explained that in this transport, active transport or passive transport. Now, for the endocytosis and exocytosis, we can say we can explain it in terms of vesicle-mediated transportation, which is a transportation of large substances which may require changes in the membrane shape and fusion of the plasma membrane with vesicles that contain substance that are going to be moved. So these changes occur throughout the lifetime of the cell, and movement of materials of the cell involve changes of the membrane, formation of vesicles, which is called exocytosis, while movement of materials into the cells is called what? Endocytosis. So for exocytosis, materials can be exported from the cell by fusing vesicle with the plasma membrane. So this is an example of exocytosis. So you are transporting, this is the vesicle that is transporting materials outside, exporting materials outside of the cell. So this is cell membrane. This is cell membrane. Now, this vesicle now come and fuse to the cell membrane, causing the cell membrane to deform. You know, the shape will change so that this can empty out this content outside the cell. So this is exocytosis. While for endocytosis, there's this movement of substances, okay, inside the cell. So this needs the cell membrane to form these vesicles, and this vesicle will now be removed, and it will now come as the vesicle inside the cell with the substances that are needed for the cell to be inside the cell. So endocytosis, you can have endocytosis process in the cell, example of which include pinocytosis. When you hear pino, it's like it's involved fluid, means when the vesicle here entrap or engulf fluid, it's fluid or other macromolecules that are needed for inside the cell. You can say it is pinocytosis. You can have receptor-mediated endocytosis, whereby the molecules that are going to be inside the cell have to attach themselves to receptors on the cell membrane before they are being transported inside. And we have we have another one which is called the phagocytosis, which is carried out by phagocytes, which are some special white blood cells that engulf bacteria or the foreign substances that are needed to be degraded by the cell. So you have that process of what? Phagocytosis. So this is what we have, and I hope you enjoyed the lecture. So please go through the materials, and also if you have any question, you can drop it in the comment section, and I'm going to look at it and also respond to your questions appropriately. So thank you for watching, and this is the end of this video. See you next. Bye-bye.